Thank you very much for your invitation. So I'm going to give a brief overview of our research on the automatic analysis of cryo electron tomography data. So as we know, the cell is the basic functional and structural unit for all living organisms. If we zoom into a subregion of a cell, cytoplasm, so we can see that it contains a lot of different macromolecules of different structures and with different spatial localization. So macromolecules are the nanomachines that governs cellular processes. So if we want to precisely understand cellular processes, it's necessary to know the native structures and the spatial organization of all macromolecules inside a single cell, and their spatial interactions with other subcellular structures. So currently, although we can get beautiful high-resolution structures of certain macromolecules complexes, there are major limits in the way we obtain such structure. So this structure is obtained using such techniques like X-ray crystallography or single particle electron microscopy. The input of such techniques uses many macromolecules with identical structures. So such macromolecules are also obtained from a protein purification process, and the protein purification process is performed on cell extracts, and the cell extracts are obtained by breaking many cells. As a consequence of such complicated process, many native macromolecular structures are destroyed or distorted. Also, once a cell is broken, we lose the spatial organization and interaction information of such macromolecules. Ideally, we want to directly visualize all structures and spatial organizations of all macromolecules together with other subcellular components inside a single cell. However, this information has been extremely difficult to obtain due to the limitations of the data acquisition techniques. Nevertheless, the recent development of cryo electron tomography technique has made such information acquisition possible. This is an illustration of how this imaging technique works. First, the samples are quickly frozen and put into a transmission electron microscopy. And we have these electron beams to pass through this sample to obtain a projection image of the sample. Then we can rotate these samples to obtain a series of projection images. And these projection images are used to construct a 3D image of the cell sample at its close to native state and with submolecular resolution. Therefore, the constructed 3D image contains both native macromolecular structure and spatial organization and their interaction with other subcellular components. And this method has become the best technique to study the structural biology in situ. So the left figure shows a slice of 3D tomogram of a subregion of the human cell. The right figures are manual segmentations of subcellular components identified by imaging experts. So we can see that such tomograms captured rich structural features of this subregion at a nanometer resolution. However, such structural complexity posed a major challenge in analyzing such 3D images. It would take an experienced cryo electron tomography experts weeks or even months to carefully inspect the subcellular structures inside a tomogram. Automation of the identification and segmentation of subcellular structures, especially macromolecules, is critical. To solve this problem, since 2008, we have been working on developing various methods for the structural pattern mining to detect novel macromolecular structures inside tomograms without relying on any external templates. So this is an example of such a structural pattern mining analysis result. The left are two slices of two 3D tomograms of two bacterial cells. And by applying our structural pattern mining methods, we are able to detect tons of different structure patterns, among which one pattern we found is very interesting. It has a structure that very well fits the crystal structure of its Cryo EL complex. There is another pattern we found that fits very well the low resolution structure of ribosome. In addition, the patterns detected in those single cells also have very distinct spatial distributions as shown in the bottom figure. The systematic recognition and recovery of such macromolecular structures is very difficult. First, there are significant imaging limits. So because the tilt angle range is limited. So because in such case, it results in missing data in the reconstructed tomographs. And also the amount of electron dose sent through the organic material sample has to be very limited. Otherwise, the radiation will damage the macromolecular structures in the sample. Low dose resulted in very noisy tomogram images. In addition, there is high structural content complexity in such 3D images. First, a cell can be treated as a very complex living machine. It is substantially more complex than any man-made systems. The native macromolecule structure inside the cell are very diverse. And secondly, sometimes the macromolecules are inside are very crowded macromolecular environment. So in order to perform certain function efficiently, certainly so far we only know the structures of very small number of macromolecules, and the native structures of vast majority of macromolecules are unknown. In addition, we also have a big data challenge. Each tomogram is a grayscale 3D image. Its original size is of 6,000 by 6,000 by 1,500 voxels, which is a total of 54 billion voxels. Nowadays, a microscope can easily capture hundreds of such tomograms within a week. These tomograms can contain tens of millions of macromolecules of very diverse size and structures. So first, I'm going to give an overview of our exploratory methods developed for deep learning-based subtomogram classification. A subtomogram is a subvolume of our 3D subimage cropped from this big 3D tomogram. And a subtomogram is likely to contain only one macromolecule. If all the subtomograms contain identical structures, then they can be directly aligned and averaged to recover the structure. However, the macromolecules inside a cell has highly diverse structures. In such a case, before averaging, we have to first systematically separate them into subsets of macromolecules of identical structures. This process is called classification. Because we need to process tens of millions of subtomograms, we need to rely on deep learning to process such a big data. As mentioned before, nowadays it is not difficult to capture hundreds of tomograms only in several days. And each tomogram can have 64 billion voxels. These tomograms could contain tens of millions of structurally highly diverse macromolecules. More importantly, these macromolecules are highly heterogeneous, like consisting of a large number of structural classes. So the key point to deal with this big data issue is to develop a solution which can efficiently and accurately separate or filter these macromolecules and divide them into structurally homogeneous subsets. This is a basic illustration of how to use convolutional neural network for such a classification. Given a subtomogram, a set of learnable convolutional kernels is applied to obtain a set of filtered images. These filtered images are further filtered using another set of convolutional kernels. The filtered images are further fed into a fully connected layer, and the output of the fully connected layer produces a number of binary predictions on whether the subtomogram is assigned to each of the structural classes. Because a subtomogram is a 3D image, we use 3D filters instead of 2D filters that are commonly used in the deep learning-based image analysis tasks. As a result of this deep learning approach has achieved a significant improvement in both speed and accuracy compared to our previously developed post-normalization based fast and coarse classification methods. Based on the above work, we published several extended work. For example, we continue developing deep learning methods for the improved subtomogram classification accuracy. Because Cryo-ET data is fast accumulating, to further increase the throughput without a significant drop of classification performance, we further developed model compression techniques to significantly increase the subtomogram classification speed. Furthermore, we observed that deep learning methods are sensitive to certain noises in our subtomogram image data. In order to solve this problem, we have developed adversarial attack and defense techniques to significantly improve the robustness of the classification against certain noise patterns. Although supervised deep learning-based subtomogram classification has achieved significant improvement in classification speed and accuracy compared to previous methods, it has some major limitations. The successful training of such a supervised deep learning models normally requires the presence of a large amount of high-quality annotated training data. However, such training data is often difficult to obtain. For a particular structure of interest, if there is a template available, one may use template search to locate the corresponding macromolecules in the training tomograms. However, such template search is computationally very intensive and it also requires laborious manual inspections and curations. If there is no template available, then the preparation of training data through detection of the macromolecules of interest will be completely rely on manual inspection, so which is even more laborious. So previously, we and others have also developed subtomogram alignment and averaging based reference-free classification or structural pattern manual methods. However, such methods rely on 3D image alignment, which is also computationally very intensive. Another major issue is that the native structure of large number of macromolecule complexes remain unknown. In such case, it may not be very practical to include such novel structures into the training data. To significantly reduce the cost of annotation needed for the supervised deep learning methods, in recent years, as a proof of principle, we have been exploring different strategies. Most of such strategies was inspired from the computer vision and machine learning fields. For example, to reduce data annotation cost, we explored the domain adaptation, domain randomization, semi-supervised learning, weakly supervised learning, active learning, and image translation and techniques. To identify novel structures, we explored few-shot learning, open-set learning, autoencoder, and deep clustering approaches. Since the annotating Cryo-ET data is very laborious and time-consuming due to the quality controls and computational cost issues, it will be beneficial to have training data from a separate data source where a large amount of annotation is readily available or can be obtained in a high throughput fashion, such as using simulation, then transfer the learned knowledge into a new data set. However, different subtomograms dataset have different image intensity distributions due to different experimental conditions. When applying trained classification model to a new dataset with different imaging conditions, the accuracy is usually very low. The phenomenon is called domain shift in across the data source prediction. We have published several methods to solve this problem, including adversarial domain adaptation and domain randomization. So domain adaptation refers to the constructing a feature structure on target domain in an unsupervised fashion, such that its feature distribution is indistinguishable from the feature distribution of the source domain. Domain randomization refers to training a predictive model on the synthetic images with widely randomized parameters. In order to further reduce the amount of annotation needed, so we also have explored the unsupervised deep learning methods to classify subtomograms for clustering. Previously, we have developed a number of deep learning methods to classify these 3D images. However, these methods are generally supervised methods. They cannot be directly used to discover new structure classes that do not exist in training data. Also, because it's usually difficult to annotate such image data, high-quality training data is often hard to obtain. So we developed an autoencoder method to map the post-normalized subtomographs into a low-dimensional space and perform unsupervised clustering. However, such method has limited discriminating ability for noisy subtomograms. To solve the above problem, we recently developed a new deep clustering approach called DISCA. So it directly learns discriminative features in an unsupervised fashion. So given a 3D tomogram, we first pick potential regions that looks like a particle. Then we use DISCA to extract image features and cluster subtomograms. After the heterogeneous set is sorted into homogeneous structural subsets, as post-processing steps, we recover the structure by averaging each subset. The discovered patterns can then be embedded into the original tomogram space for visualization purpose. DISCA is a generalization. It's a generalized expectation maximization framework. So first, the subtomograms are fed into a randomly initialized convolutional neural network, as shown in figure A. So feature vectors are extracted from the second last layer of the network, as shown in figure B. Then multiple Gaussian mixture models with different number of clusters are fed into the data of features from all input subtomograms, as shown in figure C. So the best clustering model is selected based on the Bayesian information criteria as shown in figure D. The labels of clustering are used as current estimated labels to train the neural network by gradient propagation as shown in figure E. The output of last layer of the neural network is the predicted labels to compute the gradients as shown in figure 5. So this completes one iteration. The iterative process will continue until convergence. Aggregating these steps together, we have an iterative framework alternating between the expected step and the likelihood maximization step. Here is the result on a rat neuron dataset. We detected diverse structures such as ribosome, TRIC, proteasome, calcium phosphate, and mitochondrial membrane. I want to particularly point out that the detecting macromolecules with smaller macromolecular weights, such as the TRIC and proteasome, is a major challenge facing Cryo-ET data analysis. We further explored the methods that may potentially be used for macromolecular detection and recovery by disentangled larger body transformations and semantic contents of subtomograms. So basically, disentangled representation learning is a widely discussed problem nowadays, which aims to learn data representations that has one-to-one correspondence to ground truth generating factors. In many real-life image analysis applications, including Cryo-ET and Cryo-EM, the objects of interest undergo multiple transformations that alters their visual properties while keeping the semantic content unchanged. Here is an example of this entangled representation. Left are input figures contain handwritten digits of different shape classes with the random rotation, translation, and scaling. After disentangling the shape, rotation, and translation, and scaling using our unsupervised model, we obtain the right figure that only contains shapes. The variations in rotation, translation, and scaling are largely removed. Another type of work we have been doing is about segmentation of subtomogram or tomograms. As mentioned before, the macromolecules could be inside a very crowded molecular environment. In such a case, if we simply cut out the subtomogram around these macromolecules, then the subtomogram may not only contain the macromolecule of interest, but also some neighbor structures. And the existence of such neighbor structures may bias the recovery and the classification of this macromolecular structure of interest. Therefore, we need to segment out the region occupied by this macromolecule of interest from this subtomogram. We have developed the different types of segmentation methods for cryo-ET and other biomedical images and published papers in prestigious venues such as SCV and ISMB. So this method include voxel-wise binary classification, multitask learning, one-shot learning, point cloud segmentation, weakly supervised segmentation, vision transform with CNN and feature grafting, silent C detection, and cell organism detection, 3D detection based on 2D projections, distance transform, and object detection based on cell segmentation. We have developed a novel volumeric semantic segmentation method for 3D biomedical images. In particular, in cryo-ET data, we propose the first model that only needs the image level class labels as guidance to learn accurate volumeric segmentation. The model includes two novel designs. The first is a cross-image co-occurrence learning model, which utilizes group image consensus to tackle the challenges brought by image limits. The co-occurrence map is merged with the initial seeds produced by GradCam to generate the final localization map. The second is the intervoxel affinity learning, which explores voxel relations to predict segmentation with accurate boundaries. On both simulated and real data sets, our model achieved performance comparable to fully supervised methods, but our method requires much less amount of supervision. So the next part is the subtomogram alignment average for the macromolecular structures recovery. So formally, subtomogram alignment is a process to find optimal translation and rotation so that the rotated subtomogram can maximally overlap with another subtomogram. So such alignment is very important if we want to overlay multiple copies of subtomograms containing identical structures so that we can greatly reduce the noise. However, such alignment is not trivial because alignment of two 3D images requires search in a six-dimensional parameter space, including 3 rotational angles and 3 translations. Such search is computationally very expensive. Previously, we have developed a spherical coordinate fast Fourier transform-based method for fast subtomogram alignment. It achieved 6,000 times speed up compared to exhaustive scanning-based methods. In recent years, the deep learning has significantly advanced the field of image analysis, and we recently also developed fully unsupervised deep learning and data-driven based subtomogram alignment method. Given a bunch of pairs of the input subtomograms, our neural network model first extracts the image features, then calculates the correlation of these pairs of these image features, and then use it to infer a rigid transformation. The missing wedge effect is also compensated through imputation. This model is trained in such a way that the estimated rigid transformation can maximize the overlap of all pairs of input subtomograms. This method is unsupervised method, which means the training data does not contain the ground truth, or we do not need to add the ground truth rigid transformation into the training data. But actually, this model automatically learns the optimal translation and rotation parameters through the training. And the major advantage of such a method is this method is a machine learning or data-driven method. Therefore, with more training data available, this model can learn alignment better. And once the model is learned, then the given two testing subtomographs, it can very fastly estimate the optimal region transformation. Therefore, it can achieve a more than 100 times speed up compared to our previously published geometrical fast alignment method. We further combined our alignment and clustering method and obtained improved performance on both alignment and clustering tasks. So, and then we further combined also the segmentation task. So, and benefiting from the end-to-end unified network architecture, our method achieved overall state-of-the-art performance on both the simulated and real subtomogram processing tasks. To directly benefit the Cryo ET community, we are developing an open source AI platform for Cryo ET analysis, incorporating many methods into an integrated system. The analysis system involves methods on both tomogram level and subtomogram level by both traditional geometry-based and state-of-the-art deep learning-based approaches. Currently, it contains around 40,000 lines, implementing algorithms of 23 papers we published. In summary, we believe AI techniques, image analysis, computer vision, and machine learning, in particular, those unsupervised and weakly supervised methods, will play an increasingly important role in facilitating the automatic understanding and analysis of cellular tomograms. The techniques developed for Cryo ET analysis will also help in the advance of the computer vision field. So thank you for your attention.